The thing that's very unique about the Biology REU that we run here is that we have this nice integration between the Konza Prairie LTER site and the Ecological Genomics Institute. And so between those two things, we have two sets of faculty that are at the cutting edge of ecological and genomic research and focused on mechanisms in response to environmental change. And so between those two groups, we get really diverse students, and that diversity really makes the program great.
I go to St. Olaf College in Northfield, Minnesota. St. Olaf is a small liberal arts school, so we don't have much opportunity for research like this. So my advisors pointed me to the NSF website, where I just kind of looked through the database and picked the REUs that I thought fit my personality and what I wanted to study the most. And K-State was one of the few that I chose to apply to.
It's very, very different from learning in a science classroom. So when you do a research project, you're actually really doing the science. For some students, it seems to be a completely different experience.
The main goal of that is to give undergraduate students an opportunity to do independent research, to get an idea of what research is like so they can decide if a career in science is for them. So they're working with a mentor, and that mentor is helping to supervise their project, but it really is their project.
This is a project that's sort of a small piece of a bigger ongoing set of research programs that we're doing in my lab. We designed Rachel's project to be something she could fit within 10 weeks. It's a little bit tricky. We wanted it to be something that's really her project so that it's not just something that everybody else is doing. And she's focused on understanding diversity in a particular set of plants.
So I went up to the Black Hills region in South Dakota and took samples of Phlox hoodii, which is a little low-lying prairie plant. So we collected 22 populations, I think, and I brought them back here and pressed them so that they would make nicer barium samples, and then took some silica gel samples. And with the silica gel samples, I have done flow cytometry to look at genome size, to see if there's any differences in ploidy level. So the goal of my project is to kind of get a sense of the species boundaries of Phlox hoodii. They're highly variable plants, so they can look different even between the sun and the shade. And so kind of getting a grasp of the variation of the species.
It's a great way to try your hand at research, to see if, is this something that you enjoy doing? Would you consider a career in this? Are you interested in going to graduate school and pursuing that sort of thing? But you also get to meet people from all over the country, which is really nice.
They do a lot of things like they participate in field trips where we go camping, we go on canoe trips. We also have taken them to St. Louis to look at genomic centers and baseball games and other things like that.
I had a lot of fun in Manhattan. Having the opportunity to do lab work and field work over a 10-week period has been really a good thing for me in deciding what I want to do after I graduate.